As always, welcome to the pub, where the sky is the limit but not the resolution, and where the drinks are on the house but the drinks are on your house still. I think we're a little bit in a summer mode this week. There are around Europe, many people are still on holiday. But we have a fair number of participants nevertheless, and we have a nice program, and the sun is shining, at least at the place where I am, on a pier at Helsingborg city in Sweden, with lots of seagulls and sort of background music, but I hope you can hear my voice nevertheless. Today we have the Swedish node presenting exciting things, and I'll go to the introductions shortly, but yes, so let's go to the speakers, and today we have Julia Fernandez Rodriguez and then Hans Blom from Sweden, two different locations, Gothenburg, more in the south, and Stockholm, obviously a little bit to the east and to the north. And Julia has a broad career which has taken her from Spain, where she got her PhD, and then to the Mayo Clinic in Arizona, to EMBL, to Germany, to Canada at McGill, then back to Sweden, at the end ending up to University of Gothenburg, and she was appointed the head of the Cell Imaging Core in 2017. She has a broad publication record in the field, obviously, and she has many important positions in the Swedish community, being very active in the imaging community, and quite important for the engagement of the Swedish imaging community in Euro-BioImaging. She is active in cost actions such as NUBIs and COMMOLIS. And over then to Hans Blom, who is a true expert in super resolution microscopy, studied in Sweden, Germany, Norway, and Japan. Obtained his PhD from the Royal Institute of Technology in Stockholm in biophonics, and then he worked also as a research engineer at Olympus in Tokyo, did his postdoctoral work at a familiar group with Stefan Hell, working at the Max Planck Institute in Göttingen, and very actively there working on specifically super resolution technologies and for single molecule analysis. And he's now a docent, which means a sort of associated professorship at KTH, which is the Royal Technology Center or university in Sweden, and then again working obviously with super resolution. And he has been facility head at SciLife Lab in Stockholm since 2011, and also again very active in Euro-BioImaging and in engaging the imaging community with Euro-BioImaging. So I think I'll leave the introductions there, and I guess Julia, you will start, so over to you.
Thank you, John, for the great introduction you have done from us. I don't have so much to say now. Everything is done. Not really. Well, we will try to introduce today our infrastructure here in Sweden and how we have organized it during these years. As John know very well, our infrastructure in some way born or started working, tried to organize in 2014 when they have a special application for imaging in Sweden, where many universities apply, not only the ones forming initially the NMI or the National Microscopy Infrastructure. From those we were 9 or 10 different universities with different solutions in their universities. They were chosen 4 universities. That was the Royal Institute of Technology, the KTH in Swedish, Stockholm University, Umeå University, and Gothenburg University. And that's when we actually, as I say, we started in 2014, and you see there that we were founded actually in April 2016. It actually was a very long process with a lot of good recommendations for the international committee, and also a lot of meetings with the Research Council in Sweden, until finally we were founded as the National Microscopy Infrastructure. As you see, it is actually co-financed by the participating universities. And in 2020, of course, you get money. You don't get forever. In 2019, we have to apply again, and in 2020, we expand with one more university, that is Uppsala University. And what do we do? I mean, these nodes that they are situated, as you see in the picture, very well, is really well distributed in Sweden. We go from the north and east and west. We are actually divided in technical areas in some way, from that there are the expertise, according to the expertise, to the different nodes. And of course, together with the technical areas, we also have the scientific areas and backgrounds that we have around our institutions and our facilities. In KTH, as you say, John, already, we have the super resolution and the cell profiling. A special super resolution later will be more explanations from Hans about what they're doing in the node. And they have the correlative multimodal imaging in Gothenburg. It's being one big facility with several modalities on microscopy in LN-EM, and we are actually adding the MALDI as well. We have the Intravital microscopy in Stockholm as well, in Stockholm University. The correlative electron cryotomography in Umeå, that there are also, Umeå is an important hub for Science Life Lab on the cryo-EM, but that's another infrastructure, another NMI. And of course in Uppsala, thank you to our great friends there, Carolina Walby, we have the image analysis. And that was the expansion we did in 2020. See, let's pass to the north. Just wanted to show a little bit how we are at the governance structure we have. We have the main board that they have 2 representatives from each university. And of course our director, just probably forgot to say, even so that is in the slides, the host of the National Microscopy Infrastructure is in the Royal Institute of Technology in KTH, and our director is Hjalmar Brisman. And there we also have an executive management group that's led by the heads of each of the facilities that they are in different coming from the different universities. We are also having a priority committee in case that we had to prioritize projects, and this is coming, and this is a similar committee as the Science Life Lab to don't make redundant committees. We are still a small country, even if we are the biggest one in the Nordics. It's still a small country, and then we actually try to join force with Science Life Lab in many things. And of course we have as well a scientific advisory board that is international, in this case is international experts that have already been in our facilities in 2018 to do one checking and looking how we're doing and what we can do in the future. I'm sure that soon we will have to think about to have another visit from the scientific advisory board, and hope the COVID situation will allow us to do so. Of course, our mission, as most of us here on the Euro-BioImaging, is provide expertise and open access to state-of-the-art microscopy for life science research mainly. research community mainly, but of course sometimes we also have people coming from material science and other research communities. All the nodes in general will have open access to the microscopy techniques and the expertise, and you will see that I will show and enhance as well pictures and photos for our colleagues that today are not here, all of them, but they are extremely important to make the NMI a really good infrastructure. Of course, we support our users with the planning of the experiment and the execution in some cases. In some cases, we train in the end to use the microscopes. We do help them as well, of course, in the processing analysis and in the reporting. As you see, we can go for the sample prep to the image analysis and to practically the figure for the paper. We have regular training courses and also education courses, and also competence support to local facilities as well. I mean, we try to help other facilities around us with our expertise and try to lift them and to enhance this as well. Of course, the operational of the infrastructure is relied in work package. One of them is the administrative, the quality management, and that's mainly on the KTA side and with the board. And after, of course, we have the bioimaging, informatics, and process analysis, the training and dissemination, and the technology and method development. I'm not going to explain all of them, but we have people in each of the groups, and they helping the board and all of us on the heads to also make the NMI a great free infrastructure. And we also have created from the very beginning, and in this we also help, thank you the Science Life Lab, because they already have that portal accessible, and they just help us to implement it for the web portal for the National Microscopy Infrastructure, where we have the entry point on this, our NMI nodes. It's quite similar. And we'll have to say that we also copy in some way the Euro-BioImaging, the initial Euro-BioImaging, with less steps here. That we have a pre-screening by all the heads of the different facilities, that they also check that we are having a real person enter in the portal in the registration, and we also pre-screening technically to see if it's feasible. And sometimes we might send it to the local facilities because they have enough instrumentation there, and other times we'll go to facility 1, 2, 3, 4, 5, that is the different 5 nodes we have. The facility check the feasibility in case, and from there, of course, we go the project execution. As I said, we sort of have a priority group that is we're sharing with the Science Life Lab as well. And but for the moment, we have not had the case. That's what there in terms of the long-term projects. And we're thinking about, for example, projects like CLEM, correlative projects that sometimes will take a long time. But for the that have not been necessary, but we have it there in case that we actually have the need of prioritize the groups. And this is the notes and the website. It's very simple. You put nmisweden.se and you will be in our website, and there you will have not only information about all the activities and events that the nodes are organizing every year, but also you can go to the different nodes and click there, and you will probably be directed to our own homepage in our own institutions, and will show in how we can help you. You also will have the service, and also you have the link to enter in the web portal in our website. And as I say, the Research Counseling want to have a host, and this host was the Royal Institute of Technology in Stockholm. And of course they have a lot of work to do, and that's definitely something that Hjalmar could always explain much better. But they have in one point is the maintenance of the web portal. They coordinate and support the different working groups if they need. They also coordinate the education and outreach, especially the outreach and the training, and that's together coordinated together with the working group training. And they support the user access because the web portal is there sitting in the Science Life Lab, the main server, and they are checking that the proposals and reviewing and everything is in the right way. We try to be really fast as possible. We register as soon as we know that there is a person, a real register. They register, and after that, I think the reviewing and the proposals don't take more than a week or even less than that sometimes. It's true that some of the users sometimes they actually talk to us, talk to the nodes before, and then there is already a decision taken before actually. But if there is cases, and we have some cases in where they need some help, we also are there. And if Hjalmar need another help from other heads, we actually get that information as well and can support him. And now I think I will give the voice to Hans, because we will want to show you a little bit, give a glimpse of what each of the nodes doing in Sweden. Hans, the stage is yours.
Okay, thank you. Okay, so the node at the Royal Institute of Technology, we support advanced light microscopy and proteomics, antibody-based screening, you can say. That's done both on the cellular level and also in tissue. And here you see some people that are circled in blue, and those are facility staffs that are working at KTH then. And then the other peoples are the whole network. So this was a kickoff meeting we had like 3 years ago. So if you go to the next slide, we can show you some overview of what we can, so to speak, provide as service. And to the left there, you see me and my staff scientist colleague Anna. So we are the ones supporting nanoscale biological visualization with different super resolution modalities, SIMSTED, SLM, XM, and things like that. And you see some images highlighted there, both regarding synaptonemal complexes and nuclear pore complexes. And then, as we are an advanced light microscopy node, we are also looking at single molecule dynamics. So Stefan in the center there, he uses a lot of these F techniques, FCS, fluorescence correlation, FRET, STED FCS, and things like that, trying to figure out how biomolecules move and interact and what goes on there in cells or on membranes. And then we have Stephen to the right there. So he's taking care of more fast volumetric imaging with light sheet and lattice light sheet. And as Julia said, we are also, of course, with all the data we're generating, we also help out, of course, with some image analysis. And actually there to the right, you see the director of NMI, Hjalmar, that Julia mentioned. So yes, so take us to the next slide. Yes, so this is, so to speak, the second leg at KTH, where Charlotte and her team at the Spatial proteomics facility, they use a lot of antibodies. They have basically made all the human antibodies, and then they can map that onto the cells. So they are screening where all the proteins in the body is expressed on different cell lines and things like that. So they do a huge multiplexing pipeline that you can get support with on cells. But they also do that on tissue. So they have teamed up with Akoya that has been acquired actually by SAIS now, or they work together with SAIS. So they have the possibility to also go highly multiplexed imaging on tissue. So they can simultaneously measure up to 40 different proteins. So if you want support with that kind of technology, then you can come to Stockholm, and Charlotte and her team will help you with that. So next slide, please. Yes, so this is the Intravital microscopy facility also in Stockholm, but now we are at the Stockholm University. And here we made a decision when we started NMI actually to help the node at Stockholm University, so they could be really good in intravital microscopy. So Edward and his team there, they will support you with any kind of, so to speak, in vivo imaging, mouse, plants, different biomaterial, of course also in cells. So let's see, next slide, please. Yes, and these, the facility at Stockholm University, Intravital microscopy facility, they are actually inside the newly built animal house at Stockholm University. So these people has actually been in quarantine for the last 4 to 5 years, so to speak, sitting inside this animal house. And there they can do all the imaging, all the surgery of the animal, they have the animal housing and all the quarantine and things like that. So they are well equipped to help you with experiments regarding animals and then the imaging in animals or the other species. So yes, take the next slide, please. Yes, so here is an overview of what you then get support with. Deep tissue imaging, longitudinal studies in vivo, and of course also in vitro. So you can get help with surgery, imaging, you can follow your disease model or the treatments and cell dynamics and things like that. And they have a cutting edge new instrumentation installed at Stockholm University for these imaging modalities. So they can also go into film or things like that to measure a lot of parameters. Next slide, please. Yes, so here is an example then that they look in kidney tissue, and is a kidney that has been infected actually with fungus, and then they can image both the tubular morphology and they can see the fluorescence where the fungus is and how that also are expressing and growing and infecting this system. So if you need support with this kind of modalities, then just apply and Stockholm University will help you. So next slide. Yes, now we will go to the very north. And of course, where do you do cryo-EM if not in a very cold north? Well, yes, so the facility is actually doing two things up in the north. They doing light microscopy and electron microscopy, and putting that together, of course, then becomes a focus of CLEM. So they have a joint merge node up there where the light microscopist and the electron microscopist experts are working together. And next slide, please. Yes, so here is then an overview of a CLEM expert where they, so to speak, image cells with the fluorescence, and then they also take it over to the EM and the cryo-EM microscope, where they can then get, so to speak, both functional information and structural information across scales. So if you need help with that and want to go to the cold north, dark cold north in the winter, but of course very sunny in the summer, then you come to Umeå and get support with these modalities. So over to you, Julia.
Thank you, Hans. Thank you. And yes, and just to follow up the correlation and the correlative light electron microscopy that Hans have talked about in Umeå, we can help if you don't want to go really to all the north. For some of the technologies, we can help you as well on the west coast of Sweden in Gothenburg with a bit more correlated multimodal imaging. As you see here, we have super resolution microscopy as well, electron microscopy, both SEM, functional microscopy, of course. We can help the users integrating all these different modalities, because we, the facility, have already like 14 different instruments that could allow you to do so. And we have, of course, the experts that interact in between them, all that centralizing a single facility. And one of the last things we have adding in the facilities, and years ago, first it was the microfabrication technique, where we have 3D print technology to build microfluidics devices in case that we need for our users. And of course, one I think is also starting in the Euro-BioImaging, that is the multi-imaging mass spectrometry. And of course, when you have all these different type of technologies and correlated multimodal, you need a lot of imaging process analysis, because that's the way actually to integrate all these technologies, and we can help the users with those, with the image analysis and the processing. And that's the people behind the facility, divided according to their expertise. To the left side, you will see all the light microscopy people, and also the people related with the 3D printing and the microfluid devices that can running all our light microscopes and advanced systems. In the middle, we have our electron microscopies that they will help us with the different technologies and modalities as well, and to helping and making them interacted. Everything here is interacted, even so that I divided in boxes. They are not divided, and they are actually working quite as a nice thing, work. And of course, as I say, we just started now with like a year ago, less than a year ago. York, one of our main users actually, and that we have already done things with him on integrating multi-imaging mass spectrometry and light microscopy, and try to do multimodality there and correlation. He was very keen as well to make his specialist, you see here, it looks like a spaceship, his new fancy toy to be available to the users for a certain time, of course, is also for us a pilot experiment, and see how it will work, the integration of the MALDI into all our repertoire of different technologies and modalities. And this is just some examples of what we say about the correlated multimodal imaging, where we can help the people. We don't have a nanosyn here in our facility, but we have it very, very close. It's part of the natural faculty, and the machine is actually sitting in one of the hubs in AstraZeneca, and we have been helping them quite a lot on the sample preparation, and we have done a very nice, as you see here, correlation in between the transmission electron microscope and the nanoSIMs for their compounds, that they have many in Astra. And as you can imagine, on nanoSIMs, if you want to go to that resolution, they need to prepare the sample the same as the electron microscopy. And here is where our experts in electron microscopy have helped. It has not been an easy way, but it's an easy work, because it's always a problem when you prepare for EM, but in the end we have succeeded. Of course, we have mixed it as well as the other facilities, super resolution and Tokuyashu. Multimodality just by our multiphoton microscope, and in this one as well we are collaborating with Comulis in a very big project with a skin equivalence that involve more than 25 facilities and 10 countries, where the samples will go. Unfortunately, I'm not allowed to show you yet the results we get in, but they are really, really promising and very nice, where we actually doing a correlated array tomography, and of course CLEM, and as I said before, the LM MALDI imaging mass spectrometry. And I just wanted to show you, as the rest of the facilities is showing you, some example of a showcase study. I want to take one that I find it amazing and I love it. The pieces that you see here is a skin, ancient skin for 12th century mummy, and we're doing this work together with Kirsi Lawrence in Cyprus, and this is just a little piece where we have actually done different modalities already. As you see in the videos, we have a stereo, light microscopy and electron microscopy, and help of size, because we're missing the X-rays. We don't have everything. We will love it, have it, but we don't have it yet. The X-rays as well on the same tissue. Then this is where we try to do and try to integrate all these modalities and get the maximum information for the different modalities, and to answer the questions. And that's what we mean with correlated multimodal imaging node. And we have the last but not the least, it's a very important node, that is the one that we expand, and we got our colleagues on board in 2020. That is during the COVID pandemic. the bioimaging informatics facility that is sitting in Uppsala. They have a great team that is in Uppsala, and they have some people as well sitting in KTH, Kevin and Giselle. And Carolina, many of you know her very well, and she's the director of this bioinformatics facility. And of course, as you can imagine, they can help you with all what is related to analysis and as well to processing. Just to give you some examples, so what they do, I mean, this is just some examples. They have many more work to do that they do, and they can help you anything you need with analysis. They don't have microscopes, but they have experts that they can help you with your data, that going from tracking or another bacteria growth. Of course, multiplexing, now that Hans just talked before, for the facility that do multiplexing, the bioinformatics facility is actually helping them quite a lot on this tissue analysis. And of course, deep learning and machine learning, that's something that they do practically every day. And they have also one nice technique, one nice program and working where they call the tissue maps, and this is actually related as well to the multiplexing. Here you have actually the website where you can go there, and it's a combination of the spatial information that coming from RNA in situ, and together with the labeling on proteins for antibody expressions. And actually you can play there, and as you see here, you have the bases, the genes, and actually it's a quite very nice website, and they have actually as well implemented this website. That was our notes. We are very proud of all of them, and all the people that they are behind. Is an excellent team, and that's where make our infrastructure great. And of course, we will not be anyone or infrastructure if we don't have users, and we have quite many users. This is, even if it's old until 2019, it's just reflecting the amount of projects that is actually entering our facilities in total in all the nodes, and the work that we do, and all these people that we have been showing you through the presentation. And just here is a glimpse as well of many of the scientific fields where we have been helping, from neurobiology to regenerative medicine, plant biology, or even cultural heritage or food research or fuel research. Fuel research is something that we start as well here in Gothenburg, because we have the Chalmers University quite close, and we have quite a lot of researchers there, very interesting in fuel research or marine research. And of course, education. Education is extremely important. It's something that Hans as well, and we have been talking many times that we need to educate the users on what is advanced microscopy. And of course, to the left side, just wanted to show you that during this year, due to the COVID, many of our courses, or most of our courses, have been online, and we have been adapting everything to them. And not only this particular one here is in image analysis, but we have done it as well for the microscopy itself. And I want to emphasize as well in this course that is actually will happen from the 22nd to the 24th of September, where we call the NMI course or the Advanced Microscopy for Life Sciences. What is interesting in this course, of course, this time we will do online, and that will be the first time that we will do online. But this course has been running since 2017, when we did our first course. And what we have done in this course is that instead to move the trainings, we move the trainers to the trainees. Where happened, all the trainers, all the people that they are here, as you see the names, is some of the names that you already have read into the notes, and the experts in each of the fields and each of the modalities will move to some location. We have been in Linköping, in Lund, in Örebro, but unfortunately last year we have to stop. As you know why we have to stop, and we could not move our staff anymore. We thought that maybe because sometimes if we were doing the course in Gothenburg, we have only Gothenburg students, and if you do in Stockholm, only Stockholm students. We thought, okay, let's move the trainers. And of course, as you see here, say Linköping, Lund, Örebro, we have decided in the beginning to go to the universities and institutions where they are not part of the NMI, but we want to help them as well with our expertise. we are very much into education. We think that that's fundamental to people understand microscopy in general. And of course, as I said, during this year, we have been working very hard to make most of our, even not only the education of the courses, PhD courses or workshops, but also the training that we do to the microscopes. And this is very important, especially for the facility here in Gothenburg, because we normally in training give access to our instruments. Then for us it's very important, and here you see some images where you can see the people training the users in the other side, and we actually do full training in remote. But not only the training, but also the access. I mean, some of our instruments are fully automated, and that's the same for the rest of the facilities, not only from the Gothenburg facility. This is just an example, but all our systems, and most of them are automated. That means that we can do many things in remote, and that include electron microscopes as well. I want to also, just getting almost in the end, I wanted to inform you of another infrastructure that we are also part of it, that is called the Bridging Nordic Microscopy Infrastructure, the BNMI. This infrastructure was forming last year, and unfortunately the COVID have not allowed us to have many meetings and symposiums and workshops and knowledge change seminars as we thought to do it, but we have actually the first kickoff meeting, or the first on-site meeting this week. We managed to get at least one person from each of the countries, and we have actually 5 Nordic countries: Finland, Sweden, Norway, Iceland, and Denmark. And then we have got money for the next 3 years to interact more in between us, and to create a real Nordic microscopy infrastructure, a great hub. Our hub is in Norway, and where this funding that we got is mainly for organizing and symposiums and workshops and knowledge exchange. But what is important for us, and maybe for some people, if you are hearing me, is the short-term scientific mobility grants for researchers. Unfortunately, it have to be that these researchers, if they are in Europe, come to our Nordic facilities. It could be as well that maybe some of that's in the way that will work, or it will be researchers that move in between the Nordic countries, and that's the same for the facility staff. I mean, that's one of the things that we have to fulfill, that the researchers that wants to visit and they wants to use our instruments and technologies have to come to the Nordic countries, to one of the facilities in the Nordic countries. I have to also say here that we on the NMI have also a certain amount of money that we can use for some sort of scientific mobility grants, where researchers can actually come to our facilities. It's not a lot of amount of money, but we have the possibility to use that money to help in the researchers on the mobility, especially for the traveling and the hotel. And important just happened next week, and you still have time to register for the Comulis. As John have said, we and the NMI as well, we have a lot of interaction with the other European COSAS organizations or networks, and one of them is the Comulis, the Correlative Multimodal Imaging. Of course, as I say, Gothenburg is very much into the correlative multimodal imaging, and we are organizing, unfortunately not on site. That was the idea, to do it on site, but due to the COVID as well, we have to do it online. You can visit our website, the Comulis, and you still have time to register. It's for free, the event, and I think we have a very nice group of line of speakers and contribution talks. And just I finalize with some summary of where we have here in Sweden. We have our national microscopy infrastructure started now in 2016. It is located across the whole Sweden. We have a very nice distributed facilities, 5 distributed facilities that we have already shown you, with a staff around 4 to 8 in each of the facility, and we are welcome you to visit our website on the NMI is nmisweden.se, and if you want to actually register for our course on the NMI that is also open and free of charge, you can visit that website and register. And with that, I want to thank you for your attention.
Thanks, Julia and Hans, for brilliant presentations. Very nice and interesting, broad spectrum of technologies, and a very nice presentation also of different applications, including also tools that you have available apart from the instruments, also antibodies and so forth. Before we go to questions, I just noticed in the participant list that here in Europe, we're in the middle of the working day. Obviously, in the United States, the day is only beginning, and Australia, Asia are at the end of the day. And I noticed that we have the full spectrum of time zones here. So good morning, United States, and good evening, Asia, Australia, whatever. That's very nice. Keep coming in and spread the word. It's nice to have people from all over the world. So let's go to discussion. Before, while people are thinking, I had two specific questions related to CLEM. I think, Julia, you mentioned the plans of combining MALDI imaging with CLEM, if I understood correctly, and Hans, also the combination of super resolution with CLEM. If you have any comments on those experience and challenges whatsoever, I hand over to you. And then in the meantime, the other participants can think of their questions.
Yeah, I can take the one from MALDI, and Hans can take the one for super resolution. Yes, we have been working on the MALDI for quite a while now. In a way, the MALDI, they always need some light microscopy to have the overview of the tissue. We have added in the picture I have shown you from our facility, there is actually fluorescent there, and the picture is from plaques, from Alzheimer patients. And then, of course, as you know, one of the main difficulties here is to overlay the MALDI and the LM and fuse the images, and the idea will be always to do this fusion, and that's where the processing and the analysis is very important on the images, to try to fuse and to give the MALDI the resolution that light microscopy have, because this is in a way where we want to do. And this is a project that is going with colleagues here, but also with other developers and computer scientists that they are also working together with us. They are not naming here because today is NMI, not. But I think one of the challenges have been in principle is the overlay and the fusion in between the two technologies. But we are working on that, and we are actually there. We have data, and they have been able to overlay some of the images that we are working, because the imaging mass spectrometry guys, they don't stop there, and because the sample is not completely destroyed, it's not like nanoscenes that the sample is pulverized. We actually use the LM and the MALDI to find the real plates and to cut the areas that are interesting with a laser microdissection, and going there and do more proteomics and get more molecule. Then there is a work in progress on that one, mainly to try to fuse and to overlay the two to give the resolution from the LM to the MALDI. But we're working on that, and it's actually quite promising. Everything is coming.
That sounds great. And we could consider having a little bit of a topical pub session sometime in the fall on
Yes.
Especially this kind of combination of MALDI mass spec imaging with other modalities, because I think in the future this will, or it is already in high request, and this is something that will be fast developing. When it comes to Hans, an additional question, just a combination with super resolution technologies with CLEM, but an additional feature there, which was touched upon by Julia, the absence of high power source X-ray. So, you know, calling it a light source, but basically an extremely high power X-ray source giving super high resolution X-ray. I think that is also something that is potentially in demand, not available very much, but I know that Hans Hertz and other people at KTH do have lots of projects in this respect. And just if you can touch upon that technology in your answer, and whether there are any hope that this would be included sometime in the future.
Okay, yeah, let's first go back to the first question regarding, so to speak, EM light microscopy, where we use super resolution microscopy on the light microscopy side. We have actually done collaboration. You know that Sweden is a very long country, so we've done collaboration with Gothenburg, where we, so to speak, prepare a sample, and then we can do EM imaging in Gothenburg, and then we can do super resolution microscopy in Stockholm. So that we can do. And then, as I told you, up in the north, where they do CLEM in Umeå, they also have a super resolution platform there. In that case, it's D-Storm based. So we are, so to speak, kind of set to help users doing this CLEM modalities, actually shipping samples all over Sweden, which can then hook up to maybe shipping sample from user all over Europe. And then coming back to the X-ray microscopy development that goes on at KTH since almost 20 years. Actually, there are technology being transferred right now as we speak from the technical university to the medical university. So we have not decided to include that in our facility, but the technology is almost that mature that it could be set up at the facility. Maybe in that case, one should do it at the Stockholm University Node, where they do the Intravital imaging and connection to that part. So it might come.
Very interesting. Thanks a lot. So the presentations are open for questions, and please just open your microphone and speak up, as I have a hard time seeing whether somebody is raising the hand.
Ulrich?
Hi, thank you very much for the nice presentations. I only have a couple of questions about the nodes themselves. And so the first question actually would be, so what's right now the utilized capacity of the individual nodes? So meaning, are you running at the limit? Do you get enough project requests, or is there a demand to maybe increase the nodes or even get new machines? And also maybe right now, I think you have 5 nodes. You mentioned you have 5. Are there also plans to maybe extend or have more nodes? And what would be the criteria in order to have more nodes? I guess right now you described that every node has a very specific service. Also, it's more leaning towards super resolution or towards other technologies. And then my last question, I know lots of questions right now. My last question would be how you're currently, or how these individual nodes are currently financed in order to provide their services. And is this like something stable for the long run? And whether you always kind of might have to acquire more third-party funding, or whether that also comes from more national entities.
I can take the finances because I do a lot of budgeting, I think, here. If you have the time during our presentation, we also put finance. I mean, initially we have certain amount of money come from the research counseling, and they have to be co-finance. If you get £1 million, you have to get £1 million in one way or another from your own institution. This is that's one of the important criteria to be a node as well. You have to be sure that there will be a co-financiation from your university, and they will willing to do that, because notice will not be possible. That's one of the main criteria for the research counseling. But if you read during the presentation, most of the others say, and these facilities are also have other finances. We have Science Life Lab as well. That's especially important for Hans. We have just became a Science Life Lab node for the correlative array tomography this year. But Hans have been in Science Life Lab for quite a long time now. And of course we have some foundations around us here in Gothenburg, Lundberg, Stockholm have their own, and they have the Kempe Foundation in Umeå. And of course, European grants and other finance. I mean, you're getting a little bit of money of everywhere. And of course, it's very important, at least when I'm talking from my side, but I think it's the same in KTH, we get finance from the university and the faculty where we're sitting, and in the Sahlgrenska Academy in Gothenburg, in the faculty. And then of course you get money to finance and all these pieces, and we're talking about here not for 1 year financiation, we talk about 4 to 5 years. That's the way they work. Of course, after 4 or 5 years you have to apply again. It's normal. They have to prove that you have been working and have done a good job, and then start again to apply in the next one. Then I think it's not only one place where you get the money. You actually, especially in the country, I think that will work different in Janelia Farm, but in Sweden, or I think it's the same in all the countries, you get finance for different sources, and that's the way to make it sustainable. And the fees of the users. I mean, we have fees. I mean, and no facility will be sustainable if you don't have that. Then the fees of the fees as well are making a big improvement here. Hans, you want to touch the other question?
Yeah, I can try. I might not even remember them, but becoming bigger, I don't think that's so to speak focus right now. We just expanded with the Uppsala node. So, and that came out from an international advisory board, and that board also say that the nodes, they should actually focus on the techniques that they are really good at. So then we should be seen as one big infrastructure helping, of course, to start with Swedish researcher with those technologies. But that can now be offered on the international scale too. So we will not copy and become 5 nodes in Sweden doing the same thing. We will complement each other. And so we will probably not expand, at least not in this period, which ends 2024. And then we will see what the evaluation is about us.
Then I mean, we will do more in technology base. I guess that's where Hans, we need to have an extra technology, and we have thought about the X-rays at some point as well.
Yes, of course, those thinkings are of course discussed, but I don't think we will add a special node, because that will also need to be evaluated from another university side then. But technology we might add. So Ulrike, what more questions did you have?
The very first one was about the utilization, meaning, so you have the different centers with the different foci on specific techniques, but so the question is, how can I say, are certain centers run over by request, whereas others maybe have only
yes.
potentially?
Yes, no, I think we have all been slightly growing during the years. I was 7 years ago, so I was totally alone in my facility, and now we are 4, and of course we have added different techniques to support, and I don't think I run at so to speak full capacity, because I could measure during nights, during weekends, on Christmas Eve and things like that. time on the microscope we can always find. Of course, I might work so much that I die, but maybe you can find a new staff person in that case.
No one wants that. No one wants that.
Don't do that, Hans. We don't have many experts in STED with the background you have. Don't do that, or at least pass all that information to Anna before you you decide to die.
So if you want to have numbers, maybe we could say that each year at my facility, 50 to 60 new projects are started in those technologies that I and my staff support.
And that's quite a number, I have to say. Yeah, that's quite a number. But thank you very much for the nice presentation. That was super insightful, and it was a great overview. Thank you very much.
I think Asta has a comment.
Yeah, so thank you for the very nice presentation, Julia. That was fantastic, and Hans as well. I actually had a question about the bioimage informatics node, because I mean, I wanted to understand, like, what is the proportion of the projects that they get, which are based on, let's say, the so-called internal data, which is the data which would probably be generated in the facilities around or within the same node, or external data, like somebody just coming up with their own data and asking them for help with the specifically image analysis. Because I mean, with growing data, these things become always much more complicated. So what are this proportion of projects? I wanted to understand.
I mean, the proportion, I don't know if Hans know the number. I guess the only one can answer you that it will be Carolina or Anna. But they are not only, of course, as I say, they don't have the microscopes themselves or any. And I have to say that they also have helped clinical imaging as well, not only, or preclinical. They have done quite a lot, because Uppsala is actually very big in clinical imaging. They are in the faculty of image analysis, and clinical imaging have been Uppsala and also Stockholm, a great tradition of image analysis. But they are open not only to internal, of course, you come with data, and they already have quite big processes work on the internal data, but of course they also have open to external, and that is clear, because when we apply for the Euro-BioImaging to express the interest to become a node, they have several letters of support of external users that already working with them. But the capacity and the number of users, unfortunately, I don't have. We have not have yet, because they enter in 2020 and we have to do the report, and they have not been done yet. That's why I show you the numbers for 2019. And then maybe in less than a year, or at least until April next year, I can give you the number of the exact projects they running. But I know that you see they expand a lot. I remember when they started was Carolina and Peter, and now they have Anna and Kevin and Gisela and Christine. They have to grow, and the people in the picture is the facility people, not the students, because Carolina Walby is also a professor, and she have a lot of students that I think they interact a lot in between the postdocs and the facility staff. There is a mix of computer scientists and pure analysts in the group, and even some people like Anna Ipsel, that is a microscopy initially when she was in Munich.
That's actually absolutely fantastic. I'm sure they're gonna get more and more busy.
Yeah, I'm sure. But I have to say as well that we have that node, but also each of our facilities, as Ham said, we already do it. I mean, in my facility, I have Rafael that is expert, and because we need to have on local, processing the data coming from different technologies and modalities, put it together and make it. That means that sometimes we really need a local expertise. Image analysis and bioinformatics is in each of the facilities. It's the same in UMEA due to the cryo-EM and the cryo and the tomography and the CLEM. And the same with our Intravital friends in Stockholm. I mean, each of us as well have certain amount of people there that they help. And they actually now are interacting as well as analysts, that they don't feel so lonely.
Thank you.
I have to say one thing more about the data and the samples, as Ham said. As he said, we are also and with the COVID, we have learned even more that people actually can send samples to us and we can prepare it. And we have done it even now with the skin project and brain project with the brain connectome, where they actually send the samples. We tell them how to fix it. We do the rest of the processing here, and we are able in some cases to preserve the fluorescent in case that they have to be preserved to do the claim. Then we have the expertise. I mean, you don't need to come always physically. You can also send your samples to any of us. And if you want to have super resolution, I can prepare it here and ship it to Hans. Hans do the pictures, send it to me, and we can send the data. Meaning you are not limited just only that you have to do come here. You can even send your cells, and we have cell cultures as well. I mean, there is a lot of facilities behind that helping our facilities to, and we have learned a lot now how to use more the shipping and more remotely. Sometimes if you send the sample, one of my staff can put the sample there. I think Johanna have experience to play with our CD7 from Germany. That means that and we are talking about expansion and getting, we were very lucky this year. We got money, and in Stockholm and Gothenburg we get a new ELIRA 7, one each. We will be 2 ELIRA 7s, actually adding as well to our repertoire and to the Euro-BioImaging, of course.